Today we're going to continue having a look at the second line of defense. So still looking at the innate or non-specific immune response. And today we're going to be focusing on the different proteins that are involved in our second line of defense to combat an invading pathogen or non-self antigen. Specifically from your study design, the part that this video will relate to is the focus on complement proteins and their role in the immune response.
A quick reminder to everyone that we are still looking at the innate or non-specific immunity. So that means that all of these different immune responses that we are looking at today, they will be the same regardless of what type of pathogen we're looking at. So it doesn't matter if it's, the flu virus or, someone's got chickenpox or they're experiencing a bacterial infection. This innate non-specific immune response, level 1 and level 2, or first line and second line of defense, your body will respond and your cells will respond in the same way because it is still the non-specific immune response. We're not targeting a specific pathogen, we are targeting every pathogen in the same way. All right, so a couple of things that you will see that are familiar on the screen here that we've talked about previously. First of all, we have talked about proteins before, quite heavily actually. So we're looking at a form of a protein that is produced by an organism, and its job is to complement or add to the function of those immune cells. So the immune cells that we spoke about last lesson, they can still do their job, but these complement proteins are produced during an immune response to assist those cells in being able to do their job quicker, better, faster, that sort of thing. They exist all around your body as is, but they are inactive. Okay, so they will not become active until they come into contact with a pathogen or a non-self antigen. Once that occurs, their shape changes slightly, and we see that protein now become an active enzyme and can communicate with our immune cells and cause other things to happen as well. Something else that we have also talked about before is a cascade reaction. So this is something that we spoke about during cell signaling when we were referring to hydrophilic signaling molecules. We talked about enzyme cascades, and also when we discussed apoptosis, we had our caspase cascade. Okay, so we're still looking at a cascade of reactions, but this time they are solely focused around complement proteins. So once one complement protein is activated, it then does have the ability to cause the cascade of reactions to activate many more complement proteins. And these complement proteins that we're talking about, they've essentially got 3 different roles in our immune system. They can recruit other immune cells, they can perform opsonization on a pathogen, or they can use something called their membrane attack complex, or MAC complex, to destroy a cell. We're going to look at them a little bit more closely on the next slide. So there are 3 things that our complement proteins can do within our immune system. Their main one can be to attract other immune cells. So these complement proteins, once they become active, they're obviously causing a cascade of reactions to activate more complement proteins, and eventually those complement proteins are going to come into contact with the immune cells that are in our body. By doing that, they are a chemical signal, and it is triggering those immune cells to seek out that site of infection and deal with the pathogen or the non-self antigen that has managed to breach the first line of defense. So it's encouraging those immune cells to come to the site of infection. The second thing that your complement proteins can do is something called opsonization. Now this is just a really fancy word for saying that the complement proteins will actually attach to the non-self antigens on your pathogen. So in the diagram here, you can see our pathogen is this, tablet shaped thing. And then we've got some little yellow half moon shaped objects attached to it. Those half moon shaped objects are our complement proteins. So by attaching to them, they kind of act like a beaker, not a beaker, a beacon that signals different phagocytic immune cells to come and, well, do what a phagocytic cell will do and engulf that pathogen. So in this diagram here, we can see that there is a macrophage at the bottom of the image. And on that macrophage, there are some receptors that are complementary to those complement proteins. And because that exists, that macrophage is now easily able to identify that a pathogen has been recognized within this, this organism and will automatically ingest it via endocytosis and begin that phagocytic process to destroy the pathogen, which is what we're seeing here in the last image. The third thing our complement proteins can do is they can create our MAC protein complex. That stood for membrane attack complex. I'm pretty sure it was on the previous slide if you need to double check what it stands for, but I'm just going to refer to it as our MAC complex. So this is essentially a whole bunch of these complement proteins working together to punch a hole in the side of a pathogen. Okay, so this needs to be a cellular pathogen, so something like a bacteria. So you can see here we've got our ring of proteins, our complement proteins, that have now created a massive pore in the plasma membrane of this particular pathogen. By creating this massive pore using the MAC protein complex, it actually forces the extracellular fluid surrounding the pathogen to rush inside that pathogen. And as that fluid rushes in, it actually causes the pathogen cell to explode. And obviously that destroys the pathogen. Okay, so it's kind of cool that we have these proteins in our body that can do so many amazing things. And then obviously once our pathogen has been destroyed by the MAC complex, our complement proteins can attract other immune cells, for example, our phagocytic cells, to come to that area where the pathogen has exploded and clean up any debris that is left over so that no other infections develop in the body. And interferon is the other protein that we're going to talk about. So this is a cytokine. Remember we spoke about cytokines during our cell signaling topic. It's one of the signaling molecules, or a type of signaling molecule that your immune cells can release. Now this specific one isn't necessarily always released by an immune cell. Interferon is actually released by any cell that has been infected by a virus. It does use this interferon signaling molecule to signal nearby cells and also immune cells. But the key that you need to remember here is that interferon is only ever released by cells that have been infected by a virus. So an intracellular pathogen, a pathogen that is infecting the inside of the cells. So you can see here in the diagram, this cell here has been infected by our little virus particles in orange. And by doing so, that has triggered our interferon protein to be manufactured and be released via exocytosis. We're leaving the cell, not entering, so exocytosis to release it. And the diagram is now showing you, I guess, some other things that interferon can actually trigger. So like I was saying, interferon is a chemical signaling molecule for our immune cells, so it can actually activate other immune cells, for example, natural killer cells. That's an example of an immune cell that will be activated to come to that site of infection and hopefully help stop the virus from infecting any new cells. It can also send a signal to any nearby cells that may have become infected to undergo apoptosis. If our cells that have been infected by a virus particle undergo apoptosis, we actually stop that virus particle from spreading to other cells. So that's why that's an advantage. The other thing interferon can do is that it can signal a neighboring cell that probably hasn't been infected yet to start destroying RNA and reducing its protein synthesis. So if it's doing that, it's hopefully slowing down the replication process of a virus. Remember, a virus needs to hijack our replication machinery inside our cell to be able to replicate itself. It can't do it on its own. So if that cell is naturally destroying any RNA and it's stopping or reducing protein synthesis, we're no longer creating new virus particles that can then escape and infect other cells. The other thing interferon can do is it can change the plasma membrane's permeability. So it makes it really hard or more difficult for our virus to penetrate that plasma membrane and actually infect the inside of the cells. So it's kind of cool that one little protein can do all of these things. Just again reminding you, interferons, they are only released by virus infected cells. Okay, so only virus infected cells produce interferon proteins.